Well, first, thank you very much for having me here. And I was asked by the three ladies over there sitting there to say calorifer. There we go. Inside joke. Okay, I hope I put the joke, the talk together to be a little bit scientific, but also to give an overview of the science behind it. Okay, quick disclaimer. So I start with this slide because I will run out of time at the end, which I can. So this is not an up-to-date picture of my research team in Sydney and some of the funding sources that support the work that we do in the lab. So I like to start with a big picture to kind of position us. And I know that in this audience, I actually don't have to probably do it, but nonetheless, a lot of people don't realize that the actual impact of genetic disease. People don't realize that the genetic diseases affect more people than HIV and cancer combined. There's over 10,000 diseases, and every year over 8 million children are born who are affected by those diseases. And the saddest part of the statistics is that about 30% of them will not live long enough to celebrate their 5th birthday. So it's a major global problem that a lot of people don't fully appreciate. But there's exciting times. We've recently witnessed 3 very important revolutions: recombinant DNA, where we can move DNA around, DNA and RNA sequencing, which we can now read the genome very rapidly, which obviously helps with diagnosis, and more recent genome engineering revolution, where we can go into the genome and change it at will. So we can go and cure the diseases right at the source. But the question is, what is the actual impact of all this progress in science and diagnostic? And I like the simplified graphic slide that I took from Professor Alexander that shows that our knowledge is growing exponentially. Our ability to diagnose diseases follows very similar path. But when it comes to actually using this knowledge to prevent or treat diseases, there's this gap that has formed. And I believe that that's where gene therapy has a perfect space to actually make a difference. But let's start with a little bit of a biology refresher. I know this has been done earlier today, but I think it's good to put everyone on the same page. So most of you probably know that all information in our bodies are written in our DNA, our genes. The gene encodes protein, and protein is like the effector. It's actually what has the action. Let's say it helps to build bonds. If you have a mutation in the gene, you have a broken gene, you have a broken protein, and then you have a lack of function of a disease. Now, human genome is written with 4 letters, A, T, C, and G. There's about 3 billion letters. Sorry.
I found it for the online people.
Okay. There's about 3 billion letters. That's about 1,000 Bibles. So if you imagine this really long sequence written with 4 letters only, the most difficult sequence out there written with 4 letters, and a single typo, a substitution, deletion, any mistake makes all the difference, or could make all the difference and change the life of a child. So again, everything starts with the DNA, it's written there. RNA is the messenger in between, and then the protein is what actually has the function. And that means that depending on the type of mutation the child may have, we will use different strategy to actually try to help and treat the child. And this is oversimplified slide, but I hope it will make the point. We can divide the mutation into 3 major types: loss of function, gain of function, and dominant negative. Let's start with a dominant negative. The dominant negative is where the mutation changes the protein. It works almost like a toxin, so it actually has a negative function. And in this case, it actually would attack the positive protein. So every gene in our body exists in 2 copies on 2 chromosomes, one from mom, one from dad. And if you imagine this one here, and so if we, just for argument's sake, each gene produces the same amount of protein. So half comes from the mom, half comes from dad. If one of them is a toxic one, it actually attacks the other one, so you get 0 output. Yeah, so you have in theory half correct, but the negative half actually destroys it. This is actually probably the most difficult challenge this type of disease. So when we look at the DNA to protein track, we have 2 options. We can either work at the messenger, so at the RNA level, or we have to go back and work at the DNA level. When we look at the loss of function, so in the loss of function, one of the alleles is mutated and produces nothing. So you have again, simplified, but you have 50% output. In a gain of function, the mutated gene makes a protein, but a protein has a different function. That's something else, but it's not a negative protein. So actually you still have only 50% of the correct protein. So we can look at them in a way, the same way. In this case, you have a little bit fewer, more options. You You can actually, in some cases, provide the protein, so kind of inject the protein. We call it the protein replacement. You can still work at the RNA level. You can go and fix the gene, or you can deliver an extra copy of the gene to supplement for that missing copy, for that mutated copy. And those three strategies here is what we would put into the bucket of gene therapy. So gene therapy is a broad term that actually has many different approaches in. So gene therapy, in the way I explain it, hopefully it sounds very simple. We just have to deliver the copy of the gene that's mutated to the patient, but it's simple in principle. It's actually very complex in practice. I'm going to give you some historical perspective just to show how complicated, how long it took. So everything started in the 40s, where we learned that our genes are made of DNA. About 10 years later, we actually discovered the structure of DNA. Another 10 years, we realized that how the genetic code, how it's written with the 4 letters. And there were some other things that happened in the meantime. We were able to culture cells, the cloning technology. It took till 1990 for the first successful the first clinical trial, gene therapy clinical trial took place. So this is 99. It took another 10 years for the first successful trial to happen. And the question is why it took 10 years. We already knew what to do. We had all this knowledge, but it took 10 years. And there's many reasons, but one of the major ones, and I like this quote by who said that there was only 3 problems with gene therapy. And that's delivery, delivery, delivery. We know what to do, we know what to do it, but without the proper tools to actually get those things there efficiently and safely, we are at loss. We're not going to be able to do it. So there's many different ways we can divide delivery methods. There's physical chemical methods. So, I'm sorry, I'm pressing something wrong. Yeah, based on chemistry, I'm not going to talk about that much. And we also use viral vectors. So we take viruses and natural viruses, and we use them as vehicles to deliver the medicines to the correct cells. And it's really this time when we started using viral vectors that created this gateway for gene therapy to really take off and start having some successes. So when we look back on this timeline, we start seeing some successes, and they're all based on viral vector delivered therapies. Another thing that we realize, and this it's an ongoing lesson, is that scientists and doctors have to work together. I can develop something and I think, oh yeah, this is the greatest cure. But if the doctor doesn't want to use it, it doesn't really work. The doctor may want something, but the technology may not exist to deliver it that way. So it's really that talking between those two groups that really makes the difference. More recently, we also really started appreciating the involvement of the families. Again, and it's really that intersect between those 3 groups talking where we now see a true progress in gene therapies. So again, back to gene therapy, a simple concept. We have a gene and we're going to deliver it to the patients. We're using viral vectors, so you can think of it as the medicine is, something would be in a vial, and the vector is, it keeps going off. It actually just delivers it to the correct cells. In our work, we use one of the viruses. It's called adeno-associated virus. I'm not going to go through all the details here. The point is, it's well known. It's not pathogenic. It doesn't cause any diseases. There's many types. We can use it for different cells. It's a pretty sweet system. It's only one of many. There's other viruses, and they all play their own role. And for different diseases, we may choose to use different viruses. When we look at AAV, there's a number of benefits, and there's also a little bit of problems. So when it comes to benefits, AAV has never caused a disease. So the virus itself is not pathogenic. When we convert it into a medicine, we make it even safer. It can drive a long-term expression. So that means that in many diseases, what we aim for is one-time treatment. You treat and the child is cured, not treated, it's actually cured. Oh, sorry. Okay, I don't know how to go back. Okay, there it is. There's no strong immunological reactions. The only problem with this vector is that, or this virus, is that about 70% of people have seen it. Our bodies have actually reacted to it. That means that there is a lot of people out there with antibodies against them. And obviously that gets worse with age, because as we live our lives, we get exposed. So children are a little bit less exposed, but it gets worse. That means that for the clinical trial, in many cases, you have to be tested for the antibodies, and that is one of the exclusion criteria in many clinical trials. So there's something to be aware of. There's 8 drugs today approved using this vector system. So it's also very nice because from the regular system that they know about and they're familiar with it, it makes things way simpler for us to do. And there's a lot happening in this space. So this is a snapshot from 2024. Not everything here is AAV based. So that's but there's a lot of clinical trials ongoing. So this is your phase 1, 2 on the outside. And as you go closer into the middle, this is the marketed drug. So you see that there's few marketed here, but you see there's hundreds and hundreds of programs coming in. So this technology really has reached the level of maturity where we can now use it to benefit patients. So that creates a very unique opportunity for us to actually make a difference. And I want to quickly introduce my group, as I already mentioned, where we are. So we are located in just outside of Sydney. I know it looks kind of far. Sydney is here. We're about 30 minutes by train past the city called Parramatta. We're in a large biomedical precinct. We see our hospital system sees about 10% of the Australian population. So it's one of the, I think it's the biggest biomedical precinct in Southern Hemisphere. CMRI, the institute we work is located, that's this little building here. There's a pediatric hospital and then there's adult hospital here. Since I moved to Australia, I've been working on what I refer to as Australian Genome Therapeutic Center. It's a pretty much an idea of bringing all the expertise and infrastructure together so that we can take a patient from diagnosis all the way to treatment all on one campus. What we were missing was viral manufacturing, and that's something that got funded by our government a few years back. So that facility is coming online. So that actually we do have everything that's required to take a patient and rapidly develop new treatments through the research, manufacturing, and clinical implementation. And that's very important that we have this facility because when we look at the cost of development of clinical products, about half of the cost is manufacturing itself. It's really one of the most expensive steps. So when we look back on this map, when I zoom in here, again, this is CMRI, this little blue building here. And then right next door here, we have this new built pediatric hospital. There's a research centers here. And in this building here on the ground level, that's where the GMP facility is. So it's a fast growing biomedical center that puts us in a great position to help patients affected by CTNNB1 and other conditions. And we take full advantage of it. We have a number of programs running in the lab. Our journey actually started with Sparla. She was the first one who approached us. We learned a lot going through this difficult journey, and now we want to apply this knowledge to help other kids affected by other diseases. So that brings me to the few smiling faces of the kids we work with. Woodman is there as well. But let's talk a little bit about our strategy and bring it down to back to CTNNB1. We don't want to reinvent the wheel. It's too difficult. Those diseases are very difficult to work with to begin with. The funding is limited. We take actually drugs that companies put on the market, and we try to piggyback of those. So we make as little changes as possible to approved drug to convert it into a new medication. In this case, we take advantage of Zolgensma, which is a drug based on one of those viruses called AAV9 for spinal muscular atrophy. And what we take, this is a graphic representation of how the viral vector looks for Zolgensma, and we simply change the gene itself. So instead of encoding SMN, we encode CTNNB1. So single change, it's a big change. But when we go to the regulatory agencies, we say that all the other elements in the vector are already known, safe, it's the same vector system. It makes it significantly simpler to develop this. So I added a slide today just again to explain, just to make sure that we're on the same page, what gene augmentation, which also sometimes people call gene addition strategy is, because that's the strategy we are taking for CTNNB1. So if you take a brain, zoom in, there's a cell, and if you look at the nucleus of the cell, it doesn't look like this, but you have those two chromosomes, and one of them has a mutated copy of CTNNB1. That means that it's not producing this protein here. That means that, again, oversimplifying, but we have 50% of the functional protein. The strategy we're taking is encoding the gene in a viral vector, delivering it to the cells, so it creates that extra you can think of it as an extra chromosome that produces the extra copy of the protein, bringing it back to 100%. Yeah, we're not removing the one that's mutated, we're just adding an extra copy. So it's relatively simple, actually. I'm not going to talk much about this, the steps of the project or the data itself, because Andrea is talking right after me. But this is and I think she probably has a very similar slide. So you will see how the work progressed from designing the vectors, looking at different cassettes to find the one that's the most effective, testing it, making sure it's effective and safe, manufacturing, and now finally being at the forefront of a clinical trial. We've seen this slide today. It took a team of very dedicated people all over the world to make this happen. And we cannot say thank you enough to everyone involved. And it took a lot of fundraising. And again, I know Spela talked about this, and she's the one who definitely should talk about this the most. and other people in the room, but this would not be possible without everyone here and generous donations from people out there. I want to, towards the end of the talk, just quickly switch to talking about, again, on a broader picture, rare and what we call them N of ones disorders. And I think it's actually important because the more I listen to the talks today, the more I realize that actually we can think, yeah, there is CTNNB1, but each of the mutations in a way is like its own disease. So depending on the mutation, it may have to be treated differently. So it does become almost like an N of one. So again, there's about 10,000 rare disorders affecting millions of people globally. It's estimated that about 10% of children born today are affected by one of those conditions. The rare diseases affect less than 1 in 2,000. 85% of those affect less than 1 in 50,000, and we sometimes refer to those as ultra rare. And some conditions are known to affect a single person in the world. So again, if you think about some of the CTNN1 mutations, you can think of it in that way. And that makes it a very difficult challenge for actually developing clinical trials, because you have to develop drugs for individual patients for very small patients populations. So new solutions specifically designed for rare diseases are needed to make this happen. So this is a figure that I took from I think it's 2022 paper. What I find interesting here, that if you look at the diseases, so in green, you have the common ones, rare, ultra rare, and hyper rare. And here's the number of diseases. Let's look at it this way. It's a little better to look that way. Make it a little bit better. So yeah, so you have a frequency that the frequency goes the opposite direction. So the ones that are less frequent, they affect actually more people. So it's actually the opposite than we would think of it. A common disease, yeah, it's common, but actually there's not that many of those diseases. There's a lot of rare diseases. So the impact, the combined impact of the rare diseases is way larger than of the common diseases. But that's not how governments look at it, because obviously now you have to develop a lot of treatments to affect the population. If I just look at again, just those colors, this is now to scale. I'm going to stretch them a little bit, but it's to scale. The way I think of it, and I think that's quite obvious, is that the common diseases, that's where the big pharma plays. Obviously, that's single disease. They can make a lot of money, and that's fair. That's the sweet spot for them. Everything else, and again, it's a majority of the picture. It's what falls within academia, philanthropic, non-profit, whichever way you would name it. This is where the foundations like CTNNB1 Foundation actually makes a difference. And you can see this is the majority of the screen. It is a big fight that we are fighting. And this fight that we talk about here today is a big part of it. Everything we're learning would help other diseases. And this is already happening. So this is, I believe it's 2017 when the first treatment, N of 1 was developed. This was an ASO, so it's a strategy that works at the RNA level. And it's a very famous case, and it really started the field of N of 1s. But this is not the only example. We know a story, many of you probably know the story of Terry. This is the first parent developed AAV-based therapy. So very similar to what we're doing here for CTNNB1. And more recent, there was just last month, there was an article about gene editing. So again, those things are happening. Again, we're getting better. There's a lot of interest in it. And it's a really exciting time to be here right now, because we can actually make a difference. Also, when we look, sorry, there's some, when we look at how the diseases get developed or the drugs get developed right now, every single drug that actually hit the market in gene therapy started in academia, then was developed by small biotech company, and eventually was brought to market by a large pharma. There's this sort of the ecosystem that formed between academia, biotech, and pharma, where the knowledge goes this way. Obviously, impact is shared, and then the scale grows from biotech to pharma. And it's the whole ecosystem that's needed for those disorders, for those drugs to be developed. But I think when it comes to rare diseases, we have to break this ecosystem. It doesn't necessarily apply. I believe that all the drugs in this space are going to be developed, yeah, probably in academia or foundations, but it's going to be the philanthropic organization like CTNNB1 Foundation that are going to have to take the effort of actually testing those drugs and potentially bringing them to the market. And that actually brings me to some questions that are still outstanding. In this space, how do we actually set up, the clinical trials? How do we make sure that we enroll enough patients? In some of these diseases, there may not be enough patients to actually do a clinical trial. If there's one patient globally, what do you do? Do we bring those therapies to the market? Like, do we actually make them marketed drugs? It's very expensive. Does it even make sense? Who pays for all this? How do we make sure that those therapies are not costing $3 million a person, which is what the therapies cost right now if the pharma takes care of it? How do we make sure that they're accessible to everyone independently of where they are located and their socioeconomic status? And can we develop in such way that the wheel kind of keeps turning by itself? Can we make it self-sustainable so we don't have to fundraise and spend years trying to develop something? We know what to do. We just have to have the means to actually do it. So again, back to the problem. I told you I started with this slide. Genetic disease impacts more people than HIV and cancer combined, but I hope that this conference shows you that we are actually making a big difference in the statistics and we are making a change. Thank you.